In today's section, we shall see the fate of pyruvate. In the beginning, we told that our glucose is broken down to pyruvate, ATP, and NADH. So, the ATP which is produced, it is used for your biological activities. Whereas your NADH, it enters into your ETC cycle, that is your electron transport chain, and from there it enters into the oxidative phosphorylation reaction to produce again ATP. So a massive amount of ATP are produced in the ETC cycle. So what about your pyruvate? What happens to it? That is what we are going to see today. So pyruvate is the end product of glycolysis. If you look into this chart, you can see that glucose is converted to two molecules of pyruvate. Why two molecules of pyruvate? Because we saw in glycolysis, your DHAP will also get converted to glyceraldehyde. So what happens there? You end up with two molecules of phosphoglyceraldehyde. Therefore, we end in two molecules of pyruvate. So, to convert glucose to pyruvate, it almost takes 10 successive reactions. In the presence of oxygen, always the pyruvate will be getting converted into acetyl-CoA in the presence of the enzyme pyruvate dehydrogenase. So this occurs in the aerobic condition, that is in the presence of oxygen. Herein, the carbon dioxide is released from your pyruvate molecule and then it gets converted to acetyl-CoA. This acetyl-CoA is your starting material for your citric acid cycle or TCA cycle, or it is also called as Krebs cycle. So this entire pathway, it occurs in the mitochondria. Whereas this pathway, it occurs in the cytoplasm. So all that reaction which occurs in the mitochondria requires the oxygen, that is the aerobic condition. So in the end product, your acetyl-CoA, when it enters into the TCA cycle, there is lot of intermediate components formed, and finally what we get is the 4 molecules of carbon dioxide and water. We shall study about this in detail. In next you can see that under anaerobic condition, the pyruvate gets converted to lactate. This lactate, it occurs or lactate or lactic acid. Okay, lactate or lactic acid. You can, any of the name is used. Okay, when this lactic acid loses its proton, okay, then it becomes lactate. So lactate formation occurs naturally in a body. That is during in the skeletal muscle when you're doing the exercise, we told, and also the WBC which lacks mitochondria, cornea of your eye, lens of your eye, testis also produces lactate or lactic acid. But sometimes what happens is, if there is an underlying disease like low oxygen, that is hypoxia, if you have infection, if there is an heart failure, that time also lactate formation occurs. And this, when this condition happens, when this disease conditions happen, then medical attention is immediately required. So the conversion of pyruvate to lactate occurs by the enzyme lactate dehydrogenase, and it occurs under anaerobic condition, that is in the absence of oxygen. And this is a reversible reaction. Okay, this is a reversible reaction. Whereas when pyruvate converts to acetyl-CoA, it is an irreversible reaction. Okay, this cannot go back into pyruvate. We shall see the others here. You can look here. See here, the glucose, when it gets converted to pyruvate, that process is called as glycolysis. That is, your glucose is breaking down into pyruvate. On the other hand, if your pyruvate is getting converted to glucose, it is called as gluconeogenesis. That is the formation of glucose. So again, you can see here, this is what we spoke, pyruvate getting converted to lactic acid or lactate in the presence of lactate dehydrogenase, and this is an anaerobic reaction, that is no oxygen, in the absence of oxygen. And you can see here, this is a reversible reaction. This pyruvate can also get converted into alanine in the presence of the enzyme alanine aminotransferase. So alanine, all of you know that it is an amino acid. So this alanine aminotransferase is the enzyme which catalyzes this reaction. Now, why this pyruvate is getting converted into alanine? The pyruvate is actually present in your muscles. But if it has to enter into the liver, it cannot enter directly like that. If it wants to enter into the liver, it has to change its configuration. So it changes its configuration into alanine and then enters into the liver, and then it leads to a separate pathway which we are not of concern of now. So remember, pyruvate gets converted to alanine. Pyruvate is present in the muscles, and if it wants to enter into the liver, under this configuration it cannot enter, so that it gets converted to alanine by the catalysis of this enzyme. Another thing is, pyruvate can get converted into oxaloacetate. This is again, it occurs under aerobic condition. And where does it occur? It occurs in the mitochondria. You all know that oxaloacetate is an intermediate of the TCA cycle. So why this oxaloacetate is necessary? Oxaloacetate is necessary because it has to run the TCA cycle, and simultaneously it acts as a reactant for your gluconeogenesis. That is, conversion of your pyruvate into glucose is known as gluconeogenesis. So there oxaloacetate is required for the continuation of the reaction. Which reaction? Gluconeogenesis. So this is also a reversible reaction. But here you can see it is non-reversible. Next, you can see this is the same picture what I have put in the first slide. That is this part we are going to speak. In some organisms like the yeast, when there is an anaerobic condition, that is again in the absence of oxygen, okay, your pyruvate can get converted into acetaldehyde, and acetaldehyde in turn can get converted to ethanol. So what is happening is the pyruvate The carbon dioxide is removed. From where it has got the carbon dioxide? From the pyruvate. This is catalyzed by the reaction pyruvate decarboxylase. Decarboxylase means removal of the carbon dioxide. Okay, so this is the carboxyl group of the pyruvate which is being removed by the enzyme decarboxylase, and it gets converted to acetaldehyde. This acetaldehyde in turn it gets converted to ethanol. So what did we get? We got ethanol and we also got carbon dioxide. This is catalyzed, the acetaldehyde to ethanol is catalyzed by the enzyme alcohol dehydrogenase. So here what happens is your NADH is getting oxidized to NAD. Kindly remember this part. I shall explain it again. This is very important. So here we shall speak about the NADH. If you remember our glycolysis, the sixth step. Okay, yeah. See this step, if you remember, in the payoff phase, glyceraldehyde 3-phosphate, it is getting converted to 1,3-bisphosphoglycerate. So what is happening here? You see that NAD, okay, NAD is the oxidized form, but what it is happening, it is getting converted to NADH, that is, it is getting reduced. Now what has happened here? All the NADs are getting converted or reduced to NADH. So here there will be a depletion of NAD. But NAD is required for the continuation of your glycolysis. So how will this NAD be replenished back? That is what we are going to see here. So in the sixth step, as I told, you see here NAD, it is reduced to NADH. So when the pyruvate, when it is getting converted to lactate or when it is converted to ethanol, what happens is NADH is oxidized to NAD. Similarly here, it gets oxidized to NAD. Now what happens? This NAD are used up here for the reactions to continue. So you can see here, this is also the previous chart what I have put. Pyruvate getting converted to ethanol, okay, and pyruvate getting converted to lactate. You can see here it is a reversible reaction. Whenever the arrow marks are like this, it is a reversible reaction. So once again I shall repeat, glucose will be converted to pyruvate, ATP, NADH. ATP is used up for your biological reaction. NADH, it enters into the ETC cycle and then into phosphorylation to produce more of ATP. Whenever the glucose is converted into pyruvate, it is known as glycolysis. The reverse, wherein the pyruvate is getting converted into glucose, is known as gluconeogenesis. So we are looking into the fate of pyruvate. What is happening to the pyruvate? You can see here, pyruvate is formed how? By the breaking of the glucose. So this pyruvate, under the oxidization condition or aerobic condition, it gets converted to acetyl-CoA. This acetyl-CoA enters into the TCA cycle or citric acid cycle, which is present in the which occurs in the mitochondria, and then it produces carbon dioxide and water, and massive amount of ATP. We get a lot of amount of ATP here. Okay. So this process is occurring in the mitochondria, whereas this process is occurring in your cytoplasm. In the next step, if you see, the pyruvate can get converted to alanine in the presence of alanine aminotransferase. This is a reversible reaction. So why it has to get converted to aniline means this pyruvate is present in the muscles, and when it has to enter into the liver, pyruvate cannot enter. It will be blocked in this configuration. It cannot enter into it. Therefore, it gets converted to alanine and then it can enter into the liver. This is a separate pathway it leads to, which we are not concerned now. So another pathway is pyruvate can get converted into oxaloacetate in the presence of oxygen. So what is the enzyme involved? The enzyme involved is pyruvate carboxylase, and we have a cofactor biotin which enhances this reaction. Where does this oxaloacetate come? It has come from the pyruvate, and this oxaloacetate, it acts as a reactant for your gluconeogenesis. Okay, that is the reverse, that from pyruvate to glucose formation, gluconeogenesis. So it is acting as a reactant for that reaction. Also, it is an intermediate of the TCA cycle, one of the intermediate of your TCA cycle. And then you can see here the pyruvate will also get converted into lactic acid or lactate. This is also reversible reaction. You can see here the pyruvate, it has a carboxyl group. This carboxyl group is removed as carbon dioxide by pyruvate decarboxylase. Then it gets converted into acetaldehyde. These are the cofactors, TPP and magnesium are the, that is thymine pyrophosphate and magnesium are the cofactors. Once it has got converted into acetaldehyde, another enzyme, alcohol dehydrogenase, which has the ability to remove hydrogen, gets converted, this acetaldehyde gets converted to ethanol. So the end product what we have got is ethanol and carbon dioxide. Where does this occur? It occurs in microorganisms such as the yeast under anaerobic condition, that is absence of oxygen. So what did I get? I got ethanol and carbon dioxide. And here what is happening is, here we are just speaking about NADH. We told that, you see the reactions, okay, see this one, NADH is oxidized to NAD. Similarly, in your lactate, NADH plus is converted to NAD. In this both lactate and in ethanol, we can see that NADH is oxidized to NAD. So we know in glucose, in the 6th step, okay, when the glyceraldehyde is getting converted, what happens is the NAD is reduced to NADH. Now in our body, what happens if there is reduction in NAD? The process of converting to glucose will not happen because NAD is required. And here what is happening? It is getting decreased because it is all getting reduced to NAD. So it should get back the NAD to continue the pyruvate pathway. So from where do we get our NAD? Means both in lactate and ethanol. Here you can see when pyruvate is getting converted to lactate or pyruvate getting converted to ethanol, we see that the NADH is oxidized to NAD. It has got oxidized to NAD, and then this NAD can go back here and it can allow a cycles to, or the pathway to go forward. That is from